Welcome to the PubMed Daily Review for April 18th, 2026. Today we scanned 6,307 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, CRISPR-Cas9 mediated homology-directed repair rescues the induced bone marrow failure in Fancc -/-. Mice by Hemantha Krishnan and Nak Tung Tran from Herman B. Wells Center for Pediatric Research, Department of Pediatrics, Indiana University School of Medicine, Indianapolis, Indiana, USA. Let's break down its key contributions. Fanconi anemia is a rare but devastating genetic disorder that leads to bone marrow failure. Current treatments often involve bone marrow transplantation, but gene therapy targeting a patient's own cells is a promising alternative. While progress has been made for some types of FA, effective gene therapies for Fanconi anemia group C are still needed. To address this, researchers used a mouse model genetically engineered to lack the Fancc gene. They employed a sophisticated gene editing technique called CRISPR-Cas9, combined with AV6 delivery, to precisely insert a functional copy of the Fancc gene back into its original location within the DNA of blood stem cells. This approach aimed to restore normal gene function and assess its ability to prevent bone marrow failure. The study achieved about 20% efficiency in correcting the Fancc gene using their CRISPR-HDR system. Importantly, this correction restored Fancc gene expression to levels comparable to healthy cells. When these corrected stem cells were transplanted into mice lacking the Fancc gene, they were protected from a drug that normally causes severe bone marrow failure. In fact, 90% of the mice receiving the corrected cells survived the challenge, demonstrating a significant rescue effect. These findings are highly encouraging, demonstrating that precise gene editing with CRISPR-Cas9 can effectively correct the genetic defect underlying Fanconi anemia group C in a preclinical model. This work provides a strong foundation for developing a gene therapy for FA patients. While promising, further research will be crucial to refine the technique and ensure its safety and efficacy for potential clinical application. Up next, our second paper out of 5, Genome-edited safe and immune-evasive human pluripotent cells, potential solution for allogeneic therapies by Vivian Tam and Danny Chan from School of Biomedical Sciences, Li Ka-shing Faculty of Medicine, the University of Hong Kong, Hong Kong's Special Administrative Region of China. Let's break down its key contributions. Allogeneic cell therapies hold immense promise for treating various diseases. However, 2 major hurdles stand in the way: immune rejection by the patient and the potential for uncontrolled cell growth. This research explores a novel approach to engineer human pluripotent cells to overcome these critical challenges. To address the challenges of cell therapy, researchers engineered human embryonic stem cells. These cells were modified to evade immune detection and equipped with a safety switch. They were then transplanted into mice with human immune systems to see if these engineered cells could form tissues and survive without being rejected or growing uncontrollably. The results were striking. Grafts derived from these engineered cells survived for a remarkable 5 months in the mice. Importantly, the immune system remained responsive to new challenges, and the kill switch effectively controlled any unwanted cell growth. This demonstrates the potential for these cells to form stable, functional tissues. This study presents a significant step forward for regenerative medicine. By creating immune-evasive and controllable stem cells, we move closer to off-the-shelf cell therapies. While promising, further validation in more complex models and eventually human trials will be crucial to realize this potential. Up next, our third paper out of 5, APO4 drives microglial lipid dysregulation in Alzheimer's disease via epigenetic reprogramming of the ASCL1/LXR-alpha-H3K4Me3 axis by Lanyan Lin and Fan Lin from Geriatric Medicine, Fujian Key Laboratory of Geriatrics Diseases, Fujian Provincial Center for Geriatrics, Fuzhou University School of Medicine, Fuzhou University-affiliated Provincial Hospital, Shengli Clinical Medical College of Fujian Medical University, Fuzhou, China. Let's break down its key contributions. Alzheimer's disease is a devastating condition, and the APO4 gene variant significantly increases your risk. We know that microglia, the brain's immune cells, can become overloaded with lipids in AD. But how exactly does APO4 cause this lipid buildup, and what are the consequences? This study aimed to uncover the molecular pathway connecting APO4 to microglial lipid problems. To investigate this, The researchers used a sophisticated approach. They studied mice engineered to carry either the human APO3 or APO4 gene. Using advanced techniques like single-nucleus RNA sequencing and epigenetic analysis, they examined microglial function. They also manipulated gene expression directly in microglia and tested how well these cells could clear amyloid beta, a hallmark of AD. The results were striking. In mice with the APO4 gene, microglia showed a significant, age-dependent increase in lipid droplet accumulation, up to 3.5 times more by 15 months compared to APO3 mice. This lipid overload was linked to impaired function, as these microglia were much less efficient at clearing amyloid beta. Importantly, the study found that APO4 led to reduced expression of a key gene called ASOL1 in microglia. When they experimentally boosted ASOL1 levels, they were able to restore normal lipid processing and improve amyloid beta clearance. So, what does this mean for Alzheimer's disease? This study reveals a specific epigenetic pathway through which APO4 contributes to AD by disrupting microglial lipid metabolism. The gene ASOL1 emerges as a key player in this process. These findings open up exciting new possibilities for developing therapies that target ASOL1 to protect against APO4-driven Alzheimer's pathology. Up next, our fourth paper out of 5, Shared Genetic Architecture Between Kidney Function and Alzheimer's Disease Across Ancestries by Dia Yang and William S. Bush. Let's break down its key contributions. We know that people with chronic kidney disease often have a higher risk of developing Alzheimer's disease. But what's going on at the genetic level? Are there shared genes that contribute to both conditions? This research aimed to answer that question by looking at genetic data from diverse populations. To tackle this, the researchers used a sophisticated analytical framework. They analyzed genome-wide association study data from over 1.5 million people for kidney function and a large number for Alzheimer's disease. They employed a range of advanced genetic techniques, including linkage disequilibrium score regression, polygenic risk score analysis, and Mendelian randomization, to pinpoint shared genetic influences across different ancestries. Interestingly, while there was no overall genome-wide genetic correlation, the study found 16 specific genetic regions, or loci, that showed shared influence in European ancestry. The APO gene was the only one identified as shared across both European and African ancestries. Most of these shared influences were due to horizontal pleiotropy, meaning different genes affect each trait, but one key variant, APOE4, showed shared causality for both conditions. So what does this mean? It tells us that the link between kidney function and Alzheimer's is complex, driven by specific genetic locations rather than a broad, genome-wide effect. The APO gene is important, but we also see significant differences in genetic architecture across ancestries. This highlights the need for future studies to explore these specific genetic pathways and develop more tailored approaches for predicting risk and potential treatments. Up next, our fifth paper out of 5, author correction, a proteogenomic atlas of 1032 brain metastases identifies molecular subtypes, immune landscapes, and therapeutic vulnerabilities by Jinyu Yang and Gao Zhang for thoracic surgery. Institute of Thoracic Oncology, West China Hospital, Sichuan University, Chengdu, China. Let's break down its key contributions. Brain metastases represent a major hurdle in cancer treatment, significantly impacting patient outcomes. To better understand these complex tumors, researchers aim to create a comprehensive molecular map. This map could reveal distinct subtypes and identify potential weaknesses for targeted therapies. This study utilized a proteogenomic approach to analyze a large cohort of 1032 brain metastases. By integrating proteomic and genomic data, the researchers aimed to uncover the intricate molecular makeup of these tumors. The goal was to identify distinct subtypes and understand the immune environment within them. The analysis of over 1,000 brain metastases yielded exciting results. Researchers successfully identified several distinct molecular subtypes, each with a unique immune profile. This detailed characterization also pointed towards specific vulnerabilities that could be exploited for new therapeutic strategies. This extensive proteogenomic atlas offers a valuable new resource for researchers and clinicians. It underscores the importance of considering the molecular diversity of brain metastases when planning treatment. Future work will focus on translating these findings into more effective, personalized therapies for patients. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.